So, how much does it cost to get a single drug to the market? Company reports estimate that on average, it takes about 2.3 billion US dollars and 15 years worth of research and development to get a single drug to the market. And on top of this, only 7.9% of all drugs that enter clinical trials ever make it out to the other side. So, in today's video, what we're going to look at is the drug discovery pipeline and how target validation fits into that, the key reasons for why drugs fail in the clinic, and how to ensure a valid target is chosen for further drug discovery to minimize failures in the clinic. So, let's dive right in. So, as you probably already know, drug discovery is a multifaceted process that requires the expertise from many different areas and disciplines, and it's often described as this pipeline. And this pipeline highlights the workflows and the key stages involved in getting a single drug to the market. So I'll quickly just introduce this pipeline concept and how target validation fits into all of that. So this drug discovery pipeline has several key stages, starting from target identification and validation, hit to lead discovery and optimization, preclinical and clinical studies, and finally, drug approval and post-market monitoring. To simply describe each of these segments, target identification is the stage where we search for potential drug targets implicated in diseases. Target validation ensures that the chosen target is suitable and safe to modulate. Hit to lead discovery identifies compounds that elicit effects on these targets, and the lead optimization tweaks the properties of the compound to make it more drug-like. These sections of the pipeline constitute what we call the discovery phase of drug development. Preclinical studies check the behavior of the compound in models and explore approaches to optimize the delivery and distribution of the drug in the body. And clinical trials test the drug product in patients to ensure they work appropriately in humans, and these stages make up the development phase of a drug discovery campaign. Finally, we get to the drug's regulatory approval and longer-term oversight in post-market monitoring to ensure long-term effects of taking the drug do not have drastic, unobserved adverse reactions. This is the commercial stage where companies begin to reap the rewards of their investments. So this drug discovery process may not always necessarily be linear, but each of these segments of the pipeline have questions and answers that need to be addressed to ensure that you have confidence going into your clinical stages of development. And the clinical stage can cost hundreds of millions of dollars, and you want to ensure that your drug molecule and your drug target have sound justification for the desired treatment. Hence the saying, fail fast, fail cheap, where you'd rather fail in the discovery stages than the clinic. So now that we have some idea of how much time, money, and effort goes into developing a single drug to get it to the market, understanding why drugs fail in the clinic can help us to identify key segments of the pipeline that can help us to address those failures. And so when we look back at the past 10 years or so of clinical trials, we see that there are 5 overarching reasons for why drugs fail. These are efficacy, safety, strategic, commercial, and operational. And so to give you some context or examples of what these terms mean, a reason that a trial might have failed due to efficacy is that the administered drug showed no significant clinical benefits over a placebo or an existing treatment. When looking at safety, it might be the case that the adverse effects of the treatment were not justified compared to the benefits that the drug offered. Strategic reasons for terminating a trial might be that a company was acquired by a competitor that was working in a similar disease area. A commercial reason might be that the cost to risk ratio might be too high to justify continuing funding the campaign. And operational failures come with coordinating the clinical trials themselves, and that might have something to do with the improper study design or poor patient recruitment. So when we take all of these reasons for failures and look at the proportions for each of them, we see that efficacy and safety make up 52 and 24% of these clinical failures, respectively. So how do we address this? Efficacy and safety are primarily addressed within the discovery phase of the drug development lifecycle. So this means ensuring that the target is safe and suitable to modulate, that the drug molecule's designed are efficacious and specific. The drug product is formulated for appropriate distribution in delivery in the body. When looking at this through the lens of target validation, it's about ensuring the target that we select aligns with the criteria that make for a good drug target. And these fall into these general categories of biological relevance, druggability, assayability, and safety. So let's look at these terms in a bit more depth and look at how each of these connect to efficacy and safety to address those key reasons for failure in the clinic. So to check the box for biological relevance, we need to essentially show that there is a clear connection between the chosen target and the disease pathophysiology. And this comes with a clear understanding of the pathophysiological mechanisms at a molecular level and how that translates to disease symptoms at a complex organism level, showing that there is a link between modulating molecular systems to the signs and symptoms shown in more complex animal models, supports the biological relevance of a chosen target. So if we take this example of a real-world case looking at the development of a therapeutic to reduce risks in heart failure, we see that there was a drug designed to inhibit the CETP enzyme, which stops the conversion of high-density lipoproteins, HDLs, to low-density lipoproteins, LDLs. And the typical biomarker that represents healthy cardiovascular function is low LDL, which the drug was able to do by inhibiting CETP function. However, the presence of high concentrations of HDL resulted in an increase in heart disease and mortality rates. So while achieving a desired biomarker profile, targeting this protein did not address other key mechanisms of the disease pathophysiology, resulting in increased risks of cardiovascular and non-cardiovascular events. So what this example shows us is that if a target isn't biologically relevant, it doesn't matter how much you change or modulate its activity, it won't result in any therapeutic benefits. By determining the biological relevance of the target, we primarily address efficacy by ensuring that there is a clear connection between the chosen drug target and the disease models and outcomes. When looking at druggability, we need to assess whether the target activity can be pharmacologically modulated. And generally, to do this, we either need to have an understanding of what kind of protein we're investigating, or we have some kind of three-dimensional structure where we can assess whether there is a binding cavity or a druggable surface that will affect its activity. So if we take some examples of potential protein targets, here we have a fatty acid binding protein which has limited binding cavities and many hydrophobic residues. And this limits the ability for specific binding interactions. And for this case, it would be difficult to develop a drug to specifically bind to and modulate the function of this target. When we look at another case, a muscarinic acetylcholine receptor, which is a GPCR, this represents a highly druggable protein where it has deep binding cavities, specific interactions that can be leveraged to activate or inactivate the protein function for effective drug therapies. By assessing the druggability of a protein target, it helps address the efficacy by ensuring that whatever drug target we select and choose to pursue, we will be able to develop a pharmacological agent that is able to effectively modulate its activity. And if we're unable to create a molecule or a biologic that can bind to our desired target, it's unlikely that we can develop any effective treatment if we continue down this road. When assessing the assayability for a drug target, we need to be able to accurately measure changes and responses within the system. To do this, we need to identify a series of biomarkers that are either relevant to the disease state or the drug target itself. And this helps validate the drug target by ensuring that we can measure and statistically compare the effects of when we modulate our proposed drug target. Furthermore, it needs to be able to be upscaled for high throughput screening so that we can compare different compounds for further drug molecule development. So if we return to the example of measuring CETP activity, we can use a fluorescence assay to directly assess its function. So in this setup, CETP transfers fluorescently labeled cholesterol esters from liposomes to LDL particles, and we track fluorescence intensity as a readout of activity. Introducing inhibitors into the assay allow us to observe their functional impact. For instance, Adding a drug, torcetrapib, should eliminate the fluorescent signal because CETP activity is blocked. However, while this assay confirms that the target performs this specific biochemical function, it tells us nothing about how this inhibition influences more complex cellular, tissue, or multi-organ systems. So to do this, we can take readings of disease-related biomarkers, such as LDL and HDL, within these more complex systems. This gives us an idea of whether there are differences between treated and untreated states. But the range of events that can modulate this measurement is now much broader and in a complex system. So having both pieces of evidence strengthen the case of causality between the activity of the target and the outcomes at this complex organism level. By knowing which biomarkers are relevant to the disease state and how they present in model systems, this helps with the translatability and understanding of how efficacious targeting this protein will be in a clinical setting. Safety, in the sense of clinical trials, can be quite broad, where toxicity and off-target effects are generally the key issues that arise, and these are addressed through the drug molecule design itself. However, when we take a look at safety through the lens of target validation, it's more so about ensuring that modulating the target activity doesn't impact important physiological processes across the body. And so we can check this by addressing two questions. One, where is the target expressed across the body? And two, how is it involved in important normal physiological processes? If we look at these two different GPCRs, we see that rhodopsin receptors are primarily expressed in the eyes, where GPR108 is expressed quite broadly across the body. If we were to develop a treatment that targeted GPR108, we would likely see a lot of adverse effects across many different tissues, as this protein is likely involved in functions across all of them. So by determining where the proposed target is expressed and what functions it's involved in, we can reduce the potential for adverse reactions that impact healthy tissues. So this improves the safety profile early on, ensuring that the proposed target is not involved in these processes across the body. So being able to address these key properties with clear connections between the molecular mechanisms and the phenotypic symptoms, we can build a good case for a valid target. Gathering information on these four key areas of biological relevance, drugability, assayability, and safety across a range of molecular and cellular complex organism models will thoroughly address all of these key issues regarding safety and efficacy and reduce the risk of failures in the clinical phase of drug development. So to bring everything in this video together, drug discovery is an extremely costly and time-consuming process, and some of the biggest reasons for failure is the lack of efficacy and safety in the clinical setting. So this can be addressed across many different stages in the drug discovery pipeline, but when we look at it through the lens of target validation, this means applying the criteria for what makes a sound investigation of a chosen drug target. This is essentially showing that it has biological relevance, and the target itself is involved in important processes in the disease pathophysiology. It comes with showing that the target is druggable and can be pharmacologically modulated. We are able to identify the key biomarkers that are indicative of therapeutic efficacy, and finally that the target is safe to modulate and doesn't affect normal physiological processes. By gathering all of this information, we built a very solid foundation for the drug discovery campaign and addressed efficacy and safety for improved clinical outcomes. And it makes the time and cost invested into any drug development a prosperous venture. So this video highlights the reason why target validation is so important in the drug discovery pipeline. And in the next video, we'll look at how to approach these investigations in target validation. So thanks for listening, and I'll see you in the next one.